Okay, terrific. Okay, wonderful. So welcome everyone to today's Redbud Chapter of the California Native Plant Society and our presentation. This evening's presentation is So Many Columbines, Connecting Genetic and Ecological Factors Spurring Speciation in Columbines. Our presentation this evening is Dr. Evangeline Ballerini. She's going to speak about the genus Aquilegia. Our local Aquilegia is Aquilegia Formosa, which is a truly gorgeous, gorgeous wildflower. And we have that near us. And what we will learn from Dr. Ballerini this evening is about the amazing diversity among the Columbines, not only here in California, but throughout North America and around the world. The genus Aquilegia has undergone a rapid radiation. It involves now 70 to 80 species with diverse floral morphologies. That means what do they look like? And habitats in the past 5 to 7 million years, a mere nothing in the world of plants. She's going to tell us about the ecological factors that promote this radiation around the world, and what are the genetic factors that help facilitate this radiation, and how do these ecological and genetic factors interact to generate new species. So we're going to learn first a little bit about Dr. Evangeline Ballerini and how she came to be so fascinated by these Aquilegia. She was born and raised in a little town of 650 people on the border of Yosemite National Park. And she grew up having lots of outdoor adventures along the bank of the Merced River and in the mountains of the Central Sierra Nevada. She didn't know it but these experiences left their mark on her. And when she went to college at UC Berkeley, she originally thought she was going to be a medical doctor, but when she got a B in general chemistry, she was like, oh my gosh, it's really hard to get A's in college. So she decided, no, maybe not mid school, but she really did like biology. So she thought, oh, genetics, evolution, these are really fascinating. They've got the rules that explain about life on Earth. So because she had a hard time killing or harming animals, she gravitated towards plants. And she really found that looking at what grad students did was really fun. So she decided she'd go to grad school. So, she went to grad school back east, but then she realized, oh, wait, what I really want to study is those Aquilegia, the Aquilegia Formosa that's back in California. So she went to grad school, studied the Aquilegia Formosa, then went back to the south, went down to the south to study irises, and finally came back to California to work on adaptation and speciation in Columbines. She did a postdoc at UC Santa Barbara. And in 2019, she accepted a position as faculty at the California State University, Sacramento, where she is now. And we are so pleased to have her with us this evening. And I personally am so looking forward to having her share with us so much about Columbine. Take it away, Dr. Ballerini.
Right, thank you so much for that introduction and for inviting me to share some of my research with you all tonight. Let me get my screen share going here. All right. So, yeah, first off, I hate to break it to you, but from my introduction, you probably gathered that I'm not really a botanist, but I would call myself an evolutionary geneticist. I'm really interested in understanding how genes and evolution interact together with ecology to generate diversity. And I work in Columbine. So yeah, as a biologist, one of the things that I'm really interested in is phenotypic variations. So phenotype is an organism's set of traits, their morphology, their shape, their color, their physiology, their metabolisms, their phenology. For example, when plants flower, all of those things can have variation within populations and between species. And what I'm really interested in is how that phenotypic variation within a population can lead to variable reproductive success. For example, if you flower in the middle of winter, you're probably not going to have as many offspring as if you flower in the spring. So understanding how variation in phenotype leads to variation in reproductive success involves ecological studies. Now, that variable reproductive success, it can lead to differences in which genes get passed on, which genetic variants get passed on to the next generation. And understanding that is largely within the field of evolutionary biology. And now that genetic variation itself is often the basis of the phenotypic variation that you see in a population. And understanding how variation in genes leads to variation in phenotypes, in those shapes, morphologies, colors, et cetera, that we try to figure out by studying developmental processes. And the research that I do touches on all of these aspects in the genus Aquilegia. So another big question that a lot of biologists have is what explains patterns of diversity across the planet? So we can look at a finer scale, right? Here's two irises that are each other's closest relatives. So what happened that led to this speciation event that led to these two species? Here's an example of two Columbines that are very closely related and two Clarkia. So some of my research focuses sort of at these really fine scale levels at closely related species, whereas other aspects of my research take a step back and look at bigger patterns of diversity. For example, here are two animal lineages that have been independently evolving from each other for the same amount of time. However, this crocodilian lineage has only about 23 species, whereas in that same amount of time, the bird lineage has evolved about 10,000 species. So why is there this big difference in the number of species in these two lineages that have been evolving for the same amount of time? And it might have something to do with something that evolved in the ancestor of birds that allowed them to speciate more than crocodiles. Maybe say a key innovation like wings. So one thing that I focus on is a key innovation, which is a feature that promotes diversification by increasing ecological opportunity. And a really good example of this in flowering plants is a floral nectar spur. All right, so here are some Ranunculaceae, Delphinium, Aconitum that have evolved these floral nectar spurs. And this lineage has more species than the sister lineage that's been evolving for the same amount of time. Now, nectar spurs have evolved multiple times within the flowering plants. And in nearly every case, the lineage that has evolved nectar spurs has more species in it than the lineage that doesn't have nectar spurs. Now, the reason we think that nectar spurs lead to more speciation is that they promote these specific interactions with animal pollinators. For example, this Aquilegia siberica is bee pollinated. And there can be several different floral adaptations that might lead flowers to be pollinated by different animal pollinators. And when that happens, the genes from this species are no longer being shared with the genes of this species. Reproductive isolation arises. That's a barrier to gene flow between populations, which is a really important factor for how new species form. So again, I study Aquilegia. They're in the family Ranunculaceae, and in the subfamily Thalattoideae. Here are flowers from some genera within this subfamily. There's Semi-Aquilegia, Para-Aquilegia, Thalictrum. The white features that you're seeing here are not petals. Those are actually sepals. Many of these genera have very tiny petals. So I don't know, these little yellow bits you can see here behind the anthers, those are the petals for these plants. Some of them lack petals altogether, often like Thalictrum isopyrum. But then you get the genus Aquilegia. And you have this tremendous amount of floral variation that, again, has evolved over the maybe past 5 to 7 million years. There are about 70 to 80 species distributed across the Northern Hemisphere. And we link this burst of diversification to the evolution of a nectar spur. Now, in Aquilegia, the nectar spur is formed by petal tissue. So here's a petal. The spur is this tubular outgrowth with a nectary at the end. There's another part of the petal that we call the blade, and it's the part sticking out in front of the flower. These structures, again here, that are acting as part of the floral display are actually the sepals. So the talk I'm giving tonight is going to have 2 main parts. One is going to look at how I've tried to identify genetic variants that are important for speciation in the North American Columbines. In the second half, I'm going to look at genetics involved in forming a nectar spur and how nectar spurs form. All right, taking a step back for a second, we can look at the distribution of Aquilegia. They're distributed throughout the Northern Hemisphere. These are just some of the species that we sequenced in a paper we published in 2018. Here's Semi-Aquilegia, which is thought to be the sister species lacking nectar spurs. Semi-Aquilegia can be found in China and Japan. We think that Aquilegia arose initially in this region of Asia. There are 3 main lineages. 2 of them are the largest. One lineage that moved west through Asia into Europe and then eventually started to move back towards Asia, and another major lineage that moved over into North America. And I'm going to focus on these North American taxa tonight. So the North American radiation of Aquilegia really is linked to floral phenotypes and pollinators. So here's a tree representing species relationships of these North American species. And these nodes are color-coded based on pollinator type. So ancestrally, we think that North American Columbines had a morphology similar to what you see here in Aquilegia Brevis styla. They were bee-pollinated. Bee-pollinated taxa typically have blue and purple colors, often long blades, often curved spurs, shorter curved spurs. There have been multiple shifts from bee pollination to hummingbird pollination. So hummingbirds only occur in the Americas. They don't occur in Europe and Asia. And from this hummingbird pollinated type, we've had several shifts to hawk moth pollination. So the hummingbird pollinated types are typically red and yellow. The hawk moth pollinated types typically have long spurs and lack blue and red pigments. They're usually white or maybe light yellow. So I want to know, what are the genetic factors that contribute to reproductive isolation and speciation between some of these taxa, specifically around when we have these big shifts in pollinator type? And I'm going to start out talking about a couple of California natives. In this clade here, Aquilegia Formosa and Aquilegia pubescens. So these 2 taxa have been studied for quite a bit of time. Here's a paper that was published in 1952 by Verna Grant, who was one of the first initial plant speciation geneticists out there. And he makes note in this paper about hybridization between Aquilegia Formosa and pubescens, that the Columbines have long been noted as being a genus with little to no sterility or incompatibility between dissimilar morphological types. So even though we get all of this variation in the flowers, you can still cross just about any species of columbine. So they haven't built up any intrinsic genetic issues with mating with each other. This means the factors that are keeping them separate as separate species are likely ecological features. So we can look at some of the ecological features that are helping keep Aquilegia Formosa and Aquilegia pubescens as separate species. So in red here is the geographic distribution, part of it, for Aquilegia Formosa. Aquilegia Formosa is very widespread, occurring from as far south as Mexico up through Alaska. And there are coastal populations along the Pacific, and some taxa occurring as far west or far east as the Rockies. Aquilegia pubescens, on the other hand, only occurs at high elevations in the central Sierra Nevada mountains. All right, so these have very different geographic distributions, although they do co-occur in the eastern Sierra Nevada. These two taxa also show habitat isolation. Aquilegia formosa will grow from 0 to about 10,000 feet in elevation, and it typically grows in more developed riparian soils, wet soils near creeks and whatnot. So all these little red flecks in here are Aquilegia formosa. Aquilegia pubescens, on the other hand, grows between 9 and 11,000 feet in very poorly developed rocky soils. So this is my dog Soda showing off sitting next to this Aquilegia pubescens plant, and all scattered throughout this rocky hillside here, this rocky mountain, are these plants. So these have a bit of habitat isolation, but they will co-occur between about 9 and 10,000 feet in the eastern drainages of the Sierra Nevada. In which case, this pollinator isolation becomes a big factor to help keep them separate species. And there are a number of traits that have been looked at that influence pollination. So some of these traits include floral color. You'll notice that Aquilegia formosa has a yellow blade. Pubescens tends to have a white blade, although there can be yellow, sometimes light yellow. There's also differences in the color of the sepals and the spurs. Aquilegia formosa is typically this red-orange color, and again, Aquilegia pubescens typically lacks pigment. There are differences in the length of the petal, both the blade and the spur. They're both longer in Aquilegia pubescens, which again serve as adaptations to hawk moths, which have a really long proboscis that can access nectar in these spurs. Whereas Aquilegia formosa tends to have a shorter spur, a bit wider, that fits the beak of hummingbirds. The flowers also differ in their orientation. Aquilegia formosa hangs pendant, pointing towards the ground, whereas Aquilegia pubescens is held upright. And a number of ecological studies have been completed on these traits. For example, you can take an Aquilegia pubescens plant and you can artificially tie it so that it's pointing towards the ground, and hawk moths don't want to visit it anymore. So a bunch of these traits are helping keep pollen from flowing back and forth between these species. However, hybrids will occur in the field, most likely due to non-specific generalist pollinators that happen to visit these plants. So my big question is, particularly with these floral phenotypes, what are the genes that are controlling the variation in these traits, these phenotypes between these two species? Why is this one red? Why does this one not make pigments, for example? So I'm going to start getting into some genetics, and if that's not what you know a lot about, I'm hoping to give you a little bit of background here. So here's some things to know about the Aquilegia genome, and I'm going to compare it to the human genome just so you have a point of reference. So the number of chromosomes. Aquilegia has 7 pairs of chromosomes, 14 total. Humans have 23 pairs, 46 total. The genome size of Aquilegia, so the genome is made up of these nucleotide base pairs, A's, C's, G's, and T's. And the genome of Aquilegia consists of 300 million of these A's, C's, G's, and T's spread across these 7 chromosome pairs. The human genome has about 10 times that many base pairs. Now, this DNA, there are regions of this DNA that we call genes, sections. That genes, typically, we refer to the regions that encode a protein. So all of the cells in our bodies, all of the cells in the Columbine, all of the cells in life, they're different because they produce different proteins. All right, so we like to look at the number of protein coding genes. Aquilegia has about 30,000 different protein coding genes, whereas a human has about 20. So again, it's differences in these protein coding genes that often determine the differences in phenotype of an organism. And I want to figure out which ones of these genes do that for different species of Columbine. So dust off what you know about Mendel, Gregor Mendel, the Austrian monk who is the father of genetics, you could say, because we're going to do a genetic cross. So we can take Aquilegia Formosa and cross it to Aquilegia pubescens. In this case, these two bars are demonstrating one chromosome pair. All right, so this Formosa inherited one chromosome from its mom and one from its dad. This pubescens inherited one chromosome from its mom and one from its dad. We can cross these together to generate what we call the first filial generation, F1 hybrid, and it inherited one chromosome from its Formosa mom and one chromosome from its pubescens dad. We can self this, cross this plant to itself, and in the gametes of this plant, the ovules and the anthers where the eggs and the sperm are being made, a process called recombination happens between these two chromosomes when we reduce them down to a single chromosome. Eggs have one chromosome, sperm have one chromosome. Those are going to come back together to form an F2. All right, so these little points here are where these pairs of chromosomes recombined. So across this chromosome, there's different patterns of inheritance. All right, we call this the genotype. So this top part up here, this F2 individual inherited 2 copies of the Formosa genes across this whole section of a chromosome. We call that homozygous for Formosa, FF, the same. In this region of the chromosome, this individual inherited 1 set of genes from Formosa and 1 set of genes from its pubescens grandparent. We call this heterozygous. It has 2 different sets of genes. Along this region, we inherit 2 pubescens copies of genes, homozygous pubescens, and then we're back to pubescens Formosa heterozygous. So this is how we would genotype across 1 chromosome pair. And I do this by sequencing genomes. So in reality, I had hundreds of these F2 individuals, and no 2 F2 individuals are going to be the same. They're all going to have slightly different combinations of chromosomes. And I'm going to sequence these individuals, sequence their genome, use some bioinformatics to figure out the genotypes. And then I'm going to associate that with different phenotypes in this F2 generation. And we get all sorts of different things. We get some red plants that are upright with long spurs. We get some pink pendant plants. We mix up all these traits that vary. Then what we want to do is look for an association between genotypes across the genome and sets of phenotypes. For example, we can look at whether or not a plant is pendant or the flowers are held upright. And if we do that, we can see here's a region of this chromosome where all the plants that are pendant are homozygous, have 2 Formosa copies of genes. Whereas that same region in all these plants that are held upright are either heterozygous or homozygous for the pubescens genes. So this is what I did for hundreds of plants. And I did it for a number of traits. I'm just going to talk about a couple here. I'm going to start with sepal and spur colored. So the difference between these taxa is that Formosa produces anthocyanin pigments. That's what give it this red color. And pubescens lacks those pigments. So I can take all of my F2s and I can look at the ones that produce anthocyanins and take measurements of that versus the ones that don't, and then look for associations with their genotype again. So this is a plot that's going to represent the genotype-phenotype association. Along the x-axis here, we have each chromosome, and it's broken up into different little bits. I sequenced the whole genome, but then I genotyped it in chunks. So each one of these is a different position on chromosome 1 where I have a genotype. On this axis, the y-axis, is going to show me how tight the association is between the genotype and the phenotype of anthocyanin production. All right, so here you can see that on chromosome 6, I have a really tight association between what these plants inherit from Formosa and pubescens and whether or not they make anthocyanins. And I can look on this, this section of the genome, and I can look at which genes are encoded there. One thing that's really nice about studying color in plants is that the biosynthetic pathways that make these colors are highly conserved across most plants. So what I'm showing you here is the anthocyanin biosynthetic pathway. Everything here in green represents an enzyme encoded by a gene, and the job of that enzyme is to process molecules through this pathway, eventually leading to these anthocyanin pigments, which give plants, again, colors from red to purple to blue. So if you break any of these enzymes, you're not going to get these colors. Now, there's another set of genes that are really important for this pathway, and those are genes that decide whether or not these enzymes get turned on in a particular tissue. So there's a set of genes that encode proteins, a WD40, something we call a basic helix loop helix, and another set of genes called R2, R3 MIBs. These proteins interact together to turn these different enzymatic genes on. All right, so we have these transcription factors and these enzymes. And we can look back at our region of chromosome 6, and I can see that there are some R2, R3 MIB transcription factors that are encoded by this region of the genome. And when I look at the sequence in my Aquilegia pubescens, my white plants, it has mutated versions of that protein that make it non-functional. So it's a really good candidate for the gene that's been targeted by evolution to create this shift from red to white. I have a method in the lab that I can use to test whether or not this gene has the function that I think it does. And I can take a wild type plant. This is a horticultural variety that we use in the lab a lot that flowers much more quickly than the wild species do. And it produces red pigment in its sepals and spurs. And I can target that gene, that MIB gene, to try to turn it down. I can't turn it all the way off. And this technique is very mishmashy. It doesn't work in all the cells all the time at the same level, but you can see that when I target it, it decreases the amount of that anthocyanin pigment in these sepals. So that's good evidence that this is the gene that was targeted by evolution for this shift important in speciation. I can look at another trait. In this case, I'll look at blade color. Again, Aquilegia Formosa made these yellow blades, whereas Aquilegia pubescens lacked those carotenoids. So carotenoids often are giving plants a yellow to orange colored pigment range. And just like the anthocyanins, the carotenoid pathway is pretty well known, pretty well characterized, and pretty well conserved across many plants. So there's a biosynthetic aspect, again, where we're making these carotenoid pigments. It requires genes to move through this whole, different enzymes to move through this pathway. Carotenoids are actually important precursors in a bunch of other different pathways that are important for plants. They're involved in photosynthesis. Aspects of it are involved in scent. So there are some genes that actually break down these carotenoids because the breakdown products are useful in other parts of the plant. So carotenoid pathway is more biosynthetic and turnover. And here's what I get when I look for phenotype, genotype associations for carotenoids. So there's a region here on chromosome 3, and it encodes a gene that makes an enzyme called carotenoid cleavage dioxygenase. And if you can't guess by the name, I'll tell you, its job is to cut up carotenoids. It's part of the turnover pathway. There's another gene over here encoding phytoene synthase. Synthase is involved in the synthesis of these carotenoids. So there are differences in how many carotenoids you might produce, as well as whether or not you're breaking down these carotenoids. And again, I can target, in this case, I targeted that carotenoid cleavage gene and tried to turn it off. So normally this wild type Aquilegia origami plant does not produce carotenoids. because this gene is cutting them up. If I turn this gene off, it can't break down those carotenoids anymore and they accumulate. So here's some blades from the knockdown versus the wild type, and you can see they are much more yellow. All right, so there's two good sets of genes that evolution is acting on to help create these new species. I'm going to jump to another species pair native to North America, Aquilegia Brevis styla and Aquilegia canadensis. And these two sister species represent a shift from bee pollination to hummingbird pollination. Aquilegia Brevis styla is pretty broadly distributed from Alaska through Canada and down into Montana and the Dakotas. Aquilegia canadensis is also very broadly distributed, ironically, not so much in Canada, but more along the southeastern United States. So just like I did with Aquilegia Formosa and Aquilegia pubescens, we've done one of these QTL crossing experiments. We crossed canadensis to Aquilegia Brevis styla. Here's what the F1 looks like. Here's what a handful of the F2s look like. And we measured a bunch of traits. We measured the blade color and length, the spur color and length. We also measured some curvature here. We wanted to figure out if we can figure out what makes these spurs curve or not. We measured some characteristics of the sepals, their length, width, and area, pistil length, and we also took some nectar measurements, volume and sugar concentration. So here you can see this little line here. Here's this nectar being produced in this spur here. Most of these, we haven't figured out which genes are acting. However, again, those color genes are really low-hanging fruit because they're highly conserved. So here we can look at sepal color, which again is going to be concerning anthocyanin production. And here we have a peak on chromosome 1. And underneath that peak is an enzyme, a gene encoding an enzyme, F3'5'H. So if we look back at this anthocyanin biosynthetic pathway, here's this enzyme. It's really important for determining whether or not molecules are going to be going down these parts of the pathway to produce red pelargonidins and cyanidins, or shifting over into this part of the pathway producing blue delphinidins. And we found that Aquilegia canadensis, the red species, doesn't have a functional version of this gene. Therefore, it can't produce delphinidins and is going to produce red pigments instead. We can look at blade color again in this cross. We have that same gene we had before, carotenoid cleavage dioxygenase on chromosome 3. And we had a couple of other genes that we think are acting here. One on chromosome 5 that is part of the synthetic pathway here, and a gene that encodes a transcription factor. Again, one of those kinds of genes that's responsible for turning on these enzymes. So kind of interesting, we have two different cases of very closely related plants, one that makes carotenoids and one that doesn't. One of the genes is the same, whereas there are a couple differences in what's controlling that between these two different groups of closely related species. So just to throw in another California native for you, this case we're not having a hummingbird shift, but we have another very closely related species to Aquilegia Formosa, Aquilegia Xemia. I'm going to throw a little question out there for you guys. Again, here's part of the Formosa distribution. If you know Aquilegia Xemia already, maybe don't throw this out in the comments, but if you don't, can anybody determine what might be different about Aquilegia Xemia based on this distribution that you see? I don't know if I can pull up my chat window. It's a serpentine endemic. So there are a number of isolating mechanisms, even though these are both pollinated by hummingbirds, they're going to differ in where they're occurring. Formosa, again, is not occurring on serpentine soils. Aquilegia Xemia is. I suppose because you guys are California Native Plant Society, presuming everybody knows what serpentine soil is. It's the state rock of California, and it's a pretty gnarly soil to grow on. It has a lot of heavy metals. So it takes special adaptations in plants to grow on it. These differ in their flowering time, at least where they kind of co-occur. Aquilegia Formosa will flower earlier, May, June. Aquilegia Xemia, even though it's growing in serpentine soils, which are normally thought of as being very dry, it grows in creeks and water seeps within the serpentine soil. So it actually has the ability to ride out the summer and flower later in the summer. There are also differences, subtle differences in the petal shape, right? So Aquilegia Xemia doesn't really produce a blade, whereas Formosa does. Formosa's spurs kind of jut a little bit more straight back, where the spurs of Xemia are kind of angled out. And hopefully this video loads. I'll just say we don't have QTL mapping yet for these two species, but it's something we'd like to add in the future. Is this video going to load? Yes, this is. This is from Justin Whittall. So here's a hummingbird pollinating Xemia. And you can see in part why these things hang pendant. The hummingbird actually lifts up these plants as it's pollinating. And when it does that, it's pushing the stamen against its stomach and getting pollen on there. So subtle differences in the shape of these spurs could result in different placement of the pollen on the body of these hummingbirds. All right, so that's a brief little overview of some of the genetics of speciation in North American Columbines and some of the type of work that I do. I'm going to shift things up a bit here, and I'm going to look at genetics involved in how nectar spurs get made, right? So first part of the talk was looking more at these recent evolutionary phenomenon in what causes species, but I want to know, how did Columbines evolve nectar spurs to begin with? This trait is important for making this genus radiate. How did they form in the first place? What kinds of genetic factors contribute to spur development and evolution? Unlike the color work I've talked to you about before, we don't have a good genetic pathway for making spurs. We don't know how they are made in any other taxa, so we have to start from scratch. And the approach I'm going to take is to use the spurless species Aquilegia calcarata. So there's one species of columbine that doesn't make spurs. It's native to montane regions of China. Now, it was initially thought that this spurless species diverged from the rest of the columbines prior to the evolution of the nectar spur. But now that we have the ability to use molecules to study species relationships, we know that Aquilegia calcarata is actually a very recently evolved species. Thus, the fact that it doesn't have nectar spurs represents a secondary loss of that trait. So its ancestor had spurs and then it got lost. So by studying the species, I'm not exactly studying how a spur evolved, but given that we know nothing about the genetics of making a spur, if I can figure out what gene got mutated to lose a spur, that'll give me an idea, one foothold in the pathway of the genetic pathway of what we need to make a spur. So again, I did this QTL mapping, we did the cross, we generated an F1, we selfed that F1 to get a bunch of recombinants, and we identified a region of the genome where the spurless F2s are homozygous calcarata, and the spurred F2s are either heterozygous or homozygous for 2 copies of the Siberica genes. And here you can see, again, we have this QTL on chromosome 3. Now, I focused in on this region of chromosome 3, where if you were homozygous Siberica or heterozygous, you make a spur. But if you are homozygous calcarata, you don't. This region encompasses 20 million base pairs and about 1,100 genes. So again, the Aquilegia genome has about 30,000 genes. So I got rid of about 29,000 of them, but I still have quite a few to try to figure out which one is causing this difference. Which one has those mutations to make spurs go away? And here I used something called comparative gene expression. So again, there's those genes called transcription factors that are responsible for turning on other genes. And I wanted to sequence all of the genes that are turned on in a spurred petal versus those that are on in a spurless petal, right? So are there some genes that are only on in a spurred petal that aren't on in this spurless petal? And rather than just focusing on these two species that I used in my cross, I added a couple of other species. That way I could look at what's common, even though these spurs are very different in their length and other attributes, what's common for making a spur versus something that doesn't make a spur. So I needed to collect petals from early developmental stages. And I looked at 5 different developmental stages. So here you can see this scale bar is half a millimeter. And at this very first developmental stage, all of these petals in these different species are really similar. They're just these little concave structures. But even by the second developmental phase that I assayed, the spurless species are starting to make this little pocket where the spur is going to grow out. And I did several stages to capture this early development of the spur. And I looked again at all the genes that are on in these tissues, and I compared what's on in these spurred ones versus off. And then I also compared that to the 1,100 genes in my QTL. And I found that there are 11 genes that are on in a calcarata on at a higher level than these guys. And I found 9 genes that are on in the spurred species at a higher level than a calcarata. And I can look at what those genes encode. We get these broad names for proteins. A lot of them are like, what? Mitogen activated protein, kinase, 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 chloroplast, beta amylase, all right? This one stuck out to me, C2H2 zinc finger transcription factor. Again, this is one of those genes that controls the expression of a lot of other genes. And in other species, relatives of this gene are known to control a developmental switch. As plants grow, they start out undergoing mitosis. To make a new floral organ, a petal, you need to make cells first. So we're dividing, we're making new cells. At some point, those cells will switch into a differentiation phase where they're going to take on some of the features that are important to that particular cell type. So this is common across plants to have this developmental switch from mitosis to differentiation. And this gene is known to control those processes in other species. So I wanted to look at what would happen, again, if I turned this gene off, if I turned it down in a spurred species. And I used my lab rat again. And again, this technique that we have to turn it down is really variable. So I also targeted one of the color pigment genes so that I would have an idea whether or not it was working. So this red petal right here is wild type. I'm not knocking down this gene. But these white petals over here all have some degree of knockdown of this gene. And you can see that they're different. There's a spur phenotype. So here's some examples of the variation I get when I target that gene for knockdown. And in the strongest cases, I'm almost completely eliminating the spur. So this gene is definitely playing a role in spur development, but I wanted to know a little bit more about how. So some prior work has been done looking at how spurs develop. A former postdoc or a former grad student in the lab that I got my PhD in did this work where he tracked petals at different times through development, and he counted the cell number in those petals. So you can see that between about 0 and 10 millimeters, right? So these little spurs, when they're growing from 0 to 10 millimeters, they're getting more and more cells. So at that point, this spur is growing by cell proliferation. But after about 10 millimeters to 40 millimeters, we're not getting any more cells. What's happening is as these cells differentiate, they start getting pigmented and whatnot, they're also getting longer. So most of the length of this spur has to do with making cells longer rather than making more cells. But we do have these 2 phases, this early phase where we're making cells and this second phase where we're elongating cells. This former grad student, Josh Puzzi, he also looked at what is causing differences between species with different spur lengths. And he found that longer spurs, this Aquilegia longissima, have longer cells than, say, this cerulean type has a smaller cell, and then this canadensis and vulgaris type have even smaller cells. So what's creating interspecies differences in length has to do with the length of the cell. So I wanted to know, my spurs, I can make them smaller by knocking down this gene. Is it affecting cell number or is it affecting cell length? Is my gene controlling cell proliferation, mitosis, or is it controlling cell shape? So I took a bunch of petals from my knockdown plants, some wild type, some with various lengths, and I sent them off to a collaborator of mine who is also a graduate student in my PhD advisor's lab, Molly Edwards, and she counted the cells and measured the lengths of these petals. And she found a very, very tight correlation with cell number and spur length. The more cells you have, the longer you are. So it looks like this gene is controlling mitosis. It's controlling the maintenance of this mitotic phase that's necessary to make a nectar spur. So in genetics, if you discover a new gene or describe a function of a new gene, you get to name it. I named this gene Popovich after Greg Popovich, who's the head coach of the San Antonio Spurs, because when Pop is on, you develop really nice nectar spurs. But if Pop is off, nectar spurs fail to develop. So again, this is more to do with losing a spur in Aquilegia calcarata, but I now want to know in the future, I want to try to figure out what was this gene doing in the ancestors of Aquilegia and its relatives that don't have spurs, and how did it gain a role in making spurs in Aquilegia? So that's what some of my future research might follow up with. So those are the stories I have to tell you about trying to figure out how genes are playing a role in speciation in Columbines. I'd like to thank members of the Hodges Lab, Scott Hodges Lab at UC Santa Barbara, where I did my postdoc, especially graduate student Nathan Derrig and several undergraduates who worked with me on projects there. My PhD advisor, Elena Kramer, as well as some of her grad students who helped me with certain aspects. Another professor at Santa Barbara, Ruth Finkelstein, some greenhouse managers at Santa Barbara who helped me maintain these F2 populations. Again, we have to grow hundreds of plants and phenotype hundreds of plants in order to conduct these studies, as well as some funding sources, the National Institutes of Health, National Science Foundation, a generous grant by Harvey Karp, and some local UC Santa Barbara grants. And with that, I will open this up to questions. If there are any.
Thank you so much, Dr. Ballerini. Oh, our minds are completely full and filled with visions of Columbines. Alonso has a question. Do you have a favorite species? And this is actually a question for you and perhaps others who might want to join in.
Do I have a favorite species? I do love working on Formosa and pubescens and their hybrids just because they're in an area that I really love going to visit. If you've ever seen Aquilegia longissima in real life, that one is pretty stunning because the spurs are about that long. So that's definitely up there as well on my list. And I do like a calcarata too. It's very dainty.
And where does longissima grow?
It is in the desert Southwest.
Oh.
I don't know, Daniel.
Yeah. So yeah, Daniel's question was, I had a question about serpentine soil chemical sequestration in relation to pigment production. Any correlations? And
I don't necessarily think so because it's the same color as the non-serpentine one. I have other projects I'm starting that I didn't really talk about here. I want to look more at nectar and nectar metabolomics, what is in nectars of different species pollinated by different pollinators. And also I've had a question about whether or not the nectar of the serpentine endemic is different, perhaps because the plant has to accumulate metals. I don't actually know how that particular species, some species can grow on the serpentine by avoiding picking up these heavy metals. Others can sequester them. And I actually don't know how Xemia does it.
Okay, fascinating. I have a question here. What would you recommend as other good places in the Western United States to go see other species of Aquilegia?
So globally, I would say that the area with the highest diversity is the Rockies, the US and Canadian. There's a lot of what we would maybe say incipient species. There's a lot of populations that might be of the same species, but that show some pretty distinct morphological differences. The state flower of Colorado, Aquilegia cerulea, you might all be aware of. And it is typically like a light blue, and it is a long-spurred species, often pollinated by hawk moths, but bees also visit it. There's quite a bit of variation in Aquilegia cerulea. There's some that don't make blue at all, they're white. And the graduate student Nathan Derricks has studied that, and he thinks it's tied to water. So if you're blue, you get warmer, you get hotter, you need more water. So potentially these blue species are occurring in parts of the Rockies that have more water, and white might be more advantageous in areas where you need better water efficiency. There's another variant of that Aquilegia cerulea that also lacks spurs, but in a different way than a calcarata. It's lost its petals completely and has a second whorl of sepals. So there's another student in Scott's lab, Zach Cabin, who's been studying that and why it might be advantageous to lose your nectar spur, given that it's so important for pollination. It looks like it has to do with herbivory, pressure to avoid herbivory. So yeah, Colorado, there's a bunch of little disjunct populations, sky islands in Nevada that can be really interesting. And because these guys hybridize, you can mix and match genes on occasion, right? These pollinators try to keep them separate, but it's not a perfect situation, so.
What are your favorite places in the Sierra to go looking not only for Aquilegia, but other plants?
One of my all-time favorites is Lundy Canyon, which, again, I grew up on the western border of Yosemite National Park, and you can take Highway 120 over the Sierras. My mom is probably hearing me say this and say, don't tell people about our favorite spot. But Lundy Canyon is up from Mono Lake, and it has gorgeous, you start out through these aspen in this canyon. It'll be Formosa territory, but if you keep hiking up this scree field, you can get towards pubescence territory, but you go through a hybrid zone. So that's one of my favorites, as well as Rock Creek in the Central Sierra as well. That's just south of Mammoth.
Okay. Yeah. Oh, it's gorgeous out there. Just totally gorgeous. Yeah.
And I will say, Catherine, who visited, I spent Sunday driving around through Sonoma County. Unfortunately, some of the local Xemia sites are in areas that got hit pretty bad with the fires last fall, the lightning complex fires. So I did find some, but some of the other areas are off limits right now because they're burnt out.
Yeah, that happens, but Yeah, they'll be back. Yeah. Yeah. Do we have any more questions? Okay. Do you have any, do you know some of the mechanisms inhibiting hybrid proliferation? Because that is a really good question.
Yeah, these hybrids, again, they will form in these eastern drainages where Formosa and pubescens co-occur. There are, when I'm out there, there's bees, there's flies, there's butterflies. So there are things around that will move pollen. And these hybrid zones are very robust. I should mention that for Formosa and pubescens, these are perennial species that I've visited the same plant for 20 years, right? So they're almost like these bushes. So these hybrid zones are pretty stable, but a former graduate student in the Hodges lab did a very nice paternity analysis study in the hybrid zones where he measured the floral traits, the ones I told you about, and he looked at seed to find out who pollinated. So if there was a plant with long spurs, but it was pink, they sequenced all these plants in this area and they could figure out who the dad was of the seed. And they knew who the mom was because they collected it off that plant. And that study showed that even within the hybrid zone, the plants that look more similar to either parental species get pollinated more and have more offspring. And they do it in a selective way where something that looks more Formosa-like is going to cross-pollinate with something that looks more Formosa-like and vice versa. So you're not really moving Formosa to pubescens or pubescens to Formosa that often. So that's what helps, that's what we think helps keep it from proliferating.
I noticed that in the general nursery trade, there is a myriad of Aquilegia hybrids.
Yep.
Yeah.
There are.
Okay.
Again, they do hybrid, like there's no internal genetic incompatibility, right? So there are some like really crossing an Asian species to a North American species, you're not going to get as much seed as you would if you were doing two North American species, but you still can get seed from just about any cross. So in gardens, in areas where maybe these haven't evolved to the ecology of a region, yeah, you get hybridization. Because of the way genetics works, I don't know if you noticed, you probably didn't, the cross I did of the Brevis styla, the bee pollinated to the hummingbird pollinated, you get all these colors, red, purple, blue in the F2 generation, but you also sometimes get white. The genetics of color is a little bit more complicated there, and you can have some bad combinations between Canadensis and Brevis styla that result in not making any pigment at all. So a lot of the growers, the horticultural industry, they create sort of something where they can cross two plants and know what they're going to get. And in F1, you pretty much know what you're going to get. But in the F2, all those traits get mixed up. So oftentimes you'll buy what you're buying from as a horticultural variety is an F1. If it goes to seed and it selfs itself, you're likely to get a whole bunch of mixes of plants in that second generation. So it's like Puggles, the dog. Like they look really cute if you get the offspring of a pug and a beagle, but if they mate with each other, you're mixing all of their genomes up at that point, and you can get all kinds of combinations that might not be as cute.
So I think this is my next question. The next question is kind of a combination of Daniel's question and the commentary about the nursery cultivars. And that is if for people who live on the urban wildlife interface, what's your view on the importance of having only the local species of the Aquilegia?
I'm not overly concerned. So one of the things, so there's one aspect that would concern me is just I'm trying to understand how evolution is happening naturally. And we do think that hybridization is a really big factor for how we've gotten so many species. Because when you hybridize, you're creating more variation, right? And you might get the variation, the combo that works the best in that environment. So we do think hybridization is really important for how all of these new species have evolved. That being said, I don't think that we are likely, these species are adapting to their environment. So if you take some genetic element that isn't adapted to that environment, it's probably not going to stick around too long. Natural selection is going to get rid of it. So I'm not generally too worried about it, in all honesty.
Okay. Okay. Good. Thank you. Okay. Any other questions? Okay. I think that's it for questions. Do you have any other final comments, Dr. Ballerini?
Not necessarily. I hope I didn't lose everybody too much with talks of genetics. I hope there were enough pretty flower pictures to hold interest a bit. But thank you for inviting me. And yeah, maybe I'll get out there and interact with you guys up the road and look at some local Formosa populations. And we do want to start this nectar biology project. So we are going to want to be collecting nectar from some of these species in the wild.
Well, if you want to have, members of the Redbud community come with you on any of those nectar gathering forays, once we can, get together as groups of people, just let Bill or Shane know, and we'll get the word out for you.
Sounds good. Thanks for inviting me again, and have a lovely rest of your Earth Day.
Thank you. Same to you. Good night, everybody. Thank you so much for coming.